which is linked with poultry and porcine viruses. And especially our theme is the strategies to combat in an Indian scenario. That's what exactly we are trying to do. We did it for poultry yesterday and we are trying to finish it for porcine today. So, and we manage it in this way. So, the word Animal Day is over here. Very interestingly, Professor Kumanan, how much we were talking about the surveillance and importance of surveillance and everything. This morning also, we were just talking about that. And come here, like the moment we talk about One Health, which this institute is leading the One Health Consortia, and when it is planned with environmental surveillance, it's really great. Thank you for choosing a wonderful title topic, and we all look forward. So I think he's already introduced what exactly we do on World Animal Day. It's an institute of animal biotechnology. So just to put another two lines, and we have been hearing from August 24th onwards the very important announced Bio E3 policy, which is linked with as a biological revolution. It has been seen, and it is linked with biotechnology for economy, environment, as well as employment. So how nice, you know. I mean, there are a couple of things which, as scientists, we can always link it with the policies. The policymakers do make it, but it makes a huge sense. And I was talking to the speaker of this day that we have important people from industry as well. And I think I saw, hi, we have a person from Rich, that's a local cluster which tags or which joins the working laboratories in One Health with PSA's office, which eventually is going to have a national One Health mission. And they are a very active unit. I'm happy that Papa gave the platform, so we got connected, and thank you for joining us. So they are kind of housed in different states. At Telangana, it's a rich uh, the entity which is connecting all of us. So moving ahead, I'll take another 1 minute to say about this Bio E3 and whatever we're doing. So when we're talking about it, One Health, zoonotic diseases, and then comes a public health, uh, importance in mind. That's what I yesterday spoke, and with that, some people can say all the viruses which are talking these 2 days symposia may not be of zoonotic importance, but it's eventually we are also concerned with food security in the coming times 2030, and then next bar is 2050. So with that, it goes very well in sync, and I'm personally very happy, and I feel good that we are going to have this lecture before we continue the brainstorm with the porcine viruses. Personally, Professor Shashidhara, well, I mean, he was here for another meeting of DBT India Alliance, and you are really kind to accept it. I know. He was doing a meeting online. He was saying, I was happy I was in car at 9:10, so I could take one meeting while traveling to NIAB, and then he says, I'll fly at 4:30. But I leave because I want to be in car, 2 o'clock, I have a meeting. Very nice for your generosity to be amongst us. I'm happy. Professor Shashidhara is known to me on screen, especially during COVID times when we were working together for inter-academia panel, and his worthy remarks and how we really have IAP has really come up very well. So with that, I personally and on behalf of NIB welcome Professor Shashidhara, and over to Yash to take it. Thank you.
Thank you, ma'am, for the wise words. Now it's my privilege to introduce our distinguished chief guest for today's World Animal Day lecture, Professor L.S. Shashidhara, Director of National Center for Biological Sciences and a leading scientist in genetics, molecular biology, and evolutionary biology. He has made significant contribution to cancer research and scientific education in India. As a founding academic member of IISER Pune and Ashoka University, he played a pivotal role in their growth. Professor Shashidhara completed his undergraduate and post-graduation studies in genetics and plant breeding at the University of Agriculture Sciences, Dharwad, and later earned his PhD from the University of Cambridge. His research group has utilized a Drosophila as a model to uncover gene regulatory mechanism that positions cells, tissues, and organs as well as to study evolutionary traits in major insect species. They have also applied these insights to investigate epithelial cancer and drug discoveries. Professor Shashidhara has been instrumental in establishing the Indian Cancer Genome Atlas and the Pune Knowledge Cluster, fostering collaborative cancer research. His leadership roles include serving as Vice President of the INSA and currently as the President of International Union of Biological Sciences, the first Indian to hold this position in its 100-year history. He has received prestigious awards, including the Shanti Swarup Bhatnagar Prize and the J.C. Bose National Fellowship, and is an elected EM board member. Well, we can carry on highlighting a few of his many accomplishments. Today, Professor Shashidhara's impact on Indian science and education is truly remarkable. Today, he will talk about environmental surveillance for One Health. Please join me in welcoming Professor Shashidhara. Please. Thank you.
Rajesh, well, I have to do a lot with you. It's thinking back.
Okay. Thank you very much for the kind introduction. Thank you very much, Dr. Sharma, for inviting me for this, important talk. To give a lecture on any, topic which I have some interest, particularly in the context of genetic evolution or development biology, is always a pleasure. But I don't know I'm qualified to talk in such an important day like World Animal Day. But I think I'll try to do a justice, because we are trying to, work, to prevent diseases of, animals, particularly livestock and poultry. So at least there is some kind of a connection for me personally in the context of World Animal Day, which is all about, for their well-being. It's not about how well we exploit them for human purposes, but it's about their own well-being for their life purposes, and not about looking from human perspective. So that's part of the One Health. Because when you say agroeconomy and all those things, everything we look at in the context of the benefit for humans. But when you look at One Health, it's not about looking at benefit for humans. It's about simply preserving the life on Earth, and life of all forms, particularly the most endangered ones, whether domesticated or otherwise. Also, they are the ones which going through a lot of stress because of increased, the pathogen load, as well as other climate change-induced stress and otherwise. So, in that connection, I'm really happy that you invited me to give this talk at the time when we started some environmental surveillance in the context of One Health. Again, I'm not qualified there are experts here from very different ICR institutions, as well as NIB itself. A large number of you are working in this area. Once you reach, in Indian context or anywhere in the world, once you reach a certain age and take certain positions, many things come to you simply because you have to manage. And manage means not necessarily that you have to understand deep science, but you should know who are the best people with whom you can get things work done, right? There are a large number of mission mode projects in the country, like Genome India projects, or this kind of One Health related mission, or Bio E3 policy now has come. There will be many more mission mode projects in the area of biotechnology. It's important that we make best use of the real expertise that are present. It may be present in a variety of different places. Thankfully, I've been able to connect some of them. They are in Bhopal, in Bhubaneswar, in Bangalore itself, or in Pune, Ahmedabad, and other places. So in a way, I'm only managing a consortium. So I'm not really a subject expert. So what I'll do is quickly, just for 3 minutes, just to my personal connection to animal. Two connection. One is I'm an animal, all of us. And second one is I do work using Drosophila as a model system. That's another connection in addition to a few other insects. So there is another connection very much. And the third is, of course, I started working on human cancer now, which is directly connected to human health.
So pointer, sir. Next slide.
Okay, thank you. So I'm a developmental biologist. My interest is how from a single cell zygote, we get large multicellular organisms. For example, a human baby which is born already has several trillion cells, right? And how, just that, how many times they're dividing to get that many trillion cells, it's all about organization and their positions and their differentiation into specific functions and all the physiological connections make one living organism. Although they each cells have their own metabolism, many cells can live on their own and function independent of other cells, like neutrophils moving around through the RBCs and trying to find the scavenger cells, trying to find where the invading bacteria is. But at the same time, each one has their own position, their own function, their own biochemical properties, and how all of these attend from a single cell. And the large understanding has come in the last 50 years, particularly post molecular biology area in 1960s and 70s, because that's when we came to know that all cells have exactly the same genome, but they actually acquire different fates during development. And that basically goes through all kinds of molecular biology, cell biology, techniques we get. Specific question that I was asking in the lab, I'm still, is how, very different organs and tissues are positioned along the body axis. You take anterior posterior axis or dorsoventral axis, very different organs and positioned. And those positioned because of a family of proteins called host proteins. And those host proteins are very important. They express in a very region-specific way. Depending on which host protein is expressed there, you get different, tissues and organs along the body axis. And within that, I'm particularly working. This is the. This is the point. Yeah. So Hox protein and one of the most studied proteins, and which is expressed in the third side axis segment of the insect, where in difference like houseflies, fruit flies, and mosquitoes, we don't have, they're only one pair of wings. That's why they're called different. The second pair, the hind wing, is modified into a balancing organ called halter. And UBX is required to make this. And if you remove UBX, you lose halter, but you gain another pair of wings. That means UBX is specify halter by modifying wing fate into an halter set. And UBX is sufficient to make halter, because if you overexpress UBX in developing wing, what you get is another pair of halter. So this was a starting point, and this is a fantastic genetic system to study multiple different kinds of questions we can ask it, which goes beyond understanding just one particular aspect of function, like what UBX does or how you get halter. For example, you can study differential growth and development, how size and shape are determined in addition to positions, right? For example, and the other one is how, as I told you, what UBX does is modifying wing fate to halter fate, right? Because if you remove UBX, what you get is a wing. So how does it do? What are the specific genes which are upregulated around it? How does it know which genes to be regulated around it? As a transcription factor, how does it know where to find? Right? What are the chemical properties of the enhancer regions? What are the chemical properties of the protein itself to do this, right? So that's the kind of question that we ask. Second one is the growth, because wing is a large structure, more number of cells, much flatter cells, whereas halter is a smaller number, they're much cuboidal or columnar cells, and they're all, form a bulbous structure. How, size and shape are determined, and that's another question that could ask. Many of things are very relevant to even human. For example, we get 2 arms and 2 legs, and 2 arms grow at exactly the same size. That means they're very highly controlled, growth control mechanisms, which basically ensure that proportionate to your body size, given a species, all your organs and tissues grow to a particular size and shape. So how does it happen during development? Again, reproducibly. If 7 billion people are there on this earth, it has happened 7 billion times in the last 50, 60 years or so, right? So that's the kind of, questions one can ask. Because of these kind of questions can be asked, we can also ask in the evolutionary perspective, For example, in insect itself, we look at all the insects which undergo what's called metamorphosis. They're holometabolous insects, like butterflies. You have a caterpillar which doesn't look like a butterfly, but when it becomes an adult, it's a beautiful butterfly, right? So this is what is called holometabolous insect. They undergo what's called a metamorphosis in their life cycle. There are 5 such insect group which undergo metamorphosis, starting from dragonflies to honeybees to beetles to moths and butterflies to flies, right? And only in flies we have 1 pair of wings. In other places there are 2 pairs. For example, in the oldest, the ancestral one is identical forewing and hindwing. If you look at any, even the color patterns, very different designs between the forewing and hindwing are identical in dragonflies, whereas it's different in case of lepidopterans. Coleopterans, there is some kind of a thick, protective sheet, whereas in honeybees, again, more or less identical forewing and hindwing. We're also trying to look at how this things has evolved over time, again, using UBX as a starting point, and variety of different experiments you can do. Using modern technology, you can take UBX from these insects, put it into Drosophila, and see whether it can rescue Drosophila mutants, or it can generate wing to halt transformation, and whether it binds to very different targets, how it binds, chromatin immunohistochemistry experiment you can do, pull down all the regulatory regions which UBX binds from different insects, compare them, and then do all kinds of transgenic experiments to verify your hypothesis and so forth. And because I was working on growth control mechanisms are during development, during stem cell, biology and regeneration are identical. It's just that stem cells are poisoned for some time, and when they get activated, they follow exactly the same growth control mechanism that happens early during development to regenerate new tissues or repair, or wound healing and so forth, right? And these stem cells can sometimes go completely crazy and then lead to tumor formation. And ultimately, some individuals may develop cancer. And turned out to be that many genes I was working in the context of growth and development in using fly model system turned out to be the either positive regulators of growth or negative regulators of growth in human. Positive regulators of growth when get overactivated, you call them as oncoproteins. The negative regulators of growth when they get suppressed, we call them as tumor suppressors. So that's how I started working with clinicians, and in fact, my entire lab now, except 1 or 2 students, are working on breast cancer in human. Anyway, so that's something about, just to say that I'm also a researcher. Didn't want to sort of come as a director here. Okay, so, today, Animal Day, I think, your director already mentioned the importance of this day. This importance, this says, again and again, we have to remind ourselves, particularly in the context of what whole world is going through, the human world as well as the animal world. And we are the ones who caused all kinds of problems for both of us. We have caused problem for ourselves, we have caused problem for them, right? And so some of the, typical standard care that we need to give to animals start with, respecting them and have compassion. And, it's nothing wrong in being, we don't need to be apologetic about using as animals and plants as part of our food. Is simply because we are part of the natural evolution and it's part of the food cycle. We are dependent on of these things. Just because we are dependent doesn't mean that we should, over utilize, graze them. Typically what happens is the way we have living in the last 10,000 years since agriculture started in human societies, our consumption has gone up beyond what ecologically one can sustain, right? For example, if you go to any forest, there are a certain number of tigers or lions you see, depending on how many of the prey for those are present. Depending on how much is the grass that is present, you will have those herbivores. So this ecological balance is well maintained in large natural system, not in the small natural system. For example, if you go to Corbett National Park, you may get large number of both herbivores and the plants for herbivores, but you may not see same many number of tigers. It's simply because of variety of different other factors which have reduced the tiger population. Sometimes it so happens that you may have large number of predators, but do not have enough herbivores for them. That also reduces. But what we have done is the huge ecological damage is by increased our consumption is simply because our population has increased, our consumption also has increased, right? Well, even domestication of animals is also sort of part of the, there are many animals which domesticate other ones. There are also predators, prey relationship, symbiotic relationship, and kind of stuff. And humans started domesticating animals before even so-called agriculture started in variety different ways. Dogs were, for example, with humans for a very long time. Then, dairy or cattle domestication started post-agriculture. Some of the wild animals are being domesticated for quite some time and so forth. But the cruelty they face from us, it should be stopped, and that should not be acceptable, right? We should consider all animals are equal. There's no point saying that one particular species or one particular type of animal is more equal than others, right? This famous animal farm story where, all animals are equal, but pigs are more equal kind of thing. I don't think there should be any such kind of a notion. We should respect all animals, irrespective of which species they belong to. Now, there is also sometimes what happens is without scientific understanding of ecology, sometimes our conservation efforts may go wrong. There is overemphasis of certain kinds of conservation, right? It's simply because they are symbols of certain kinds of societal, perceptions, right? Certain societies have certain perceptions and they try to make that particular animal much more symbolic of their societies. I'm talking about on behalf of the whole world society, not specifically Indian context. So sometimes we try to sort of conserve without going through what should be an ecological balance during the conservation. Modern understanding of genetics, ecology, and genomics, we should be able to use that for better conservation efforts. And now large data analysis is possible. You can use satellite data, you can use ground data, you can use drone capture data, and using modern data analytic methods, including artificial intelligence, the large number of good solutions can be found. Sorry. So variety of different solutions, in the context of Bio-3 policy that Dr. Sharma, you mentioned, one of the things which talking about is alternate source for protein diet. So we can reduce the, dependence on animal, protein. So animals are source of proteins, right? Most important is, reducing the infections and diseases of both domestic and wild animals. It's common to have diseases like predator-prey interaction. There's also host pathogen interaction. Having viruses and bacteria is part of our natural ecosystem. I don't think it can be avoided. Bacteria is definitely going to have a superior survival advantage than all of us, all animals and plants, simply because they divide much faster, they evolve much faster, and they have much more diverse, biochemical and physiological mechanisms to adapt to any situations, right? Given that they are going to be there, but what's happening is because of the reduced habitat and because of more close interaction among the animals, and also because of inbreeding, the genetic diversity is reduced. Invariably, they are succumbing to infectious diseases. Particularly, I'm talking about in the context of wild animals. I'm sure all of you know about recent, large, number of lion deaths in Gir forest, mostly because of reduced genetic diversity, and also they have more in a restricted zone. There is no migratory corridors which they can expand to other regions. That basically led to infection spreading much faster, right? So in a way, it's something which we need to prevent. Similarly, for domestic animals. Domestic animals are kept in close proximity in large number. If one catches into the infection, others can have infection very fast. Exactly what happened during pandemic for human. The reason earlier pandemic was not that of a problem, although we didn't have any of pharmaceutical intervention or non-pharmaceutical intervention so much without any understanding of viruses and bacteria. We managed well the COVID pandemic. I'll come to that in a minute. But still, the COVID pandemic became such a pandemic within 3 months or 4 months. It's because of a high density and high mobility of human populations, right? Second is because of the climate change, other problem we are facing, one of the problem we need to address is the methane emissions from livestock. Again, large number of livestock being grown in smaller areas and then being fed with all kinds of new types of diet, which is leading to methane emission. I am told some of you are working on new nutrition, more natural diet to reduce the methane emission from the livestock animals. Prevention is always better than cure. That's where environmental surveillance and very different surveillance, which many of our organizations are doing, we are also doing now. That's very important to prevent the outbreak. Having an early warning system, isolating the way we used to do it ourselves, test and I get isolated, right? That's how we did manage the COVID pandemic until the vaccines were available. And any pharmaceutical intervention should be more vaccine rather than trying to treat a disease, because which is anyway not going to sort of reduce the disease burden and also will lead to other complications for the animals. So non-pharmaceutical strategy or non-medicinal strategy is prevention of outbreak or using the vaccines to prevent the, at least vaccine do not prevent the infection. What vaccine will do is to prevent the disease, right? Or reduce the disease burden. So wildlife conservation, again, should be based on population genetics, genomics, and ecology. And there are more and more people are now doing this. There are a variety of different tools are available. And the cost is coming down to do all of these things. Most important, all of in Indian context is whatever we do, it should be affordable at low cost, right? So finally, as I told you, it should be based on science. That's the reason why we have set up in this country so many institutions of this kind. And NIB is one of the best in this country for animal biotechnology related work. And using deep science, deep tech, data driven, and finally the outcome should be affordable and accessible across India. That's the most important aspect of it, and we are very good in that. India is developing vaccine for the whole world, or manufacturing vaccines for the whole world, because we can actually make vaccines affordable and accessible to the masses. And that equity is most important in this country. And that's what scientists have been able to do, scientists and technocrats, with the help of academic institutions like this and industry. We've been able to do this in variety of different ways. It's not enough. We still have large number of problems to solve. Okay, so I'll sort of give some kind of overview of what we are doing with some background to this. It won't be more than 20 minutes. This purpose I've taken this talk is not only because the World Animal Day, it's also because this is the place I want you to think about it and join us in this project, and we can collaborate. And thankfully, coincidentally, we also have from ICR institutions today, particularly from Nishad in Bhopal, and that's important for our collaboration. We are anyway, collaborating in this. We already signed an MOU, and then they are visiting us on 16th next week, and we'll take this forward. So environmental surveillance for One Health. One Health, as I told you, is a health of the whole ecosystem, right? The entire chain. And there are plants, animals, and human, right? And in this ecological balance, it's very important that every individual species has its own space, and it is right in some balance. And it's not that we need to protect one over the other, and it will not be possible, right? It has to be in an ecological balance. And the only way we can do this in modern days, because of very highly dynamics, there are two problems with this we are facing in the modern world, three problems rather. One is, the most important one is the climate change induced, the problem that we have created ourselves as the humanity. The second is increased human population and increased demand for natural resources. And the third is the more political, right? All kinds of national boundaries have created us. It's separated from their fun. Whereas biodiversity and ecology, there is no, these kind of national boundaries or barriers for them, right? The pathogens move back to pollens, and seeds can move back and forth. Animals can move farther, both like birds and insects and so forth. So things can move back and forth, they maintain certain ecological balance even before the nations were born. And how to understand that ecological balance within this, because we cannot avoid having these political boundaries. We have developed in some particular way. There's no immediate solution for such kind of thing. But there should be some kind of a cooperation understanding with the help of modern data capturing and data analytic methods. We should be able to go with and One Health phenomena. One Health by default, has to be a global program. It cannot be a small regional program, right? But it has to be bottom up. So in a way, it's regional and then all the way up to the international level. So I'm sure all of you know what's One Health. I don't need to go through this. There's a standard definition as per the WHO. Now, One Health management, I just mentioned this, right? It has to be, it's for Indian context, right? So it has to be at the almost at the lowest level of the governance system. Like India, it's a panchayat system. Typically, a few villages come together under one panchayat, and it has to be from there. Right, in a city like Hyderabad, it could be a ward or it could be a small governance region, but that basically has to be looked at all aspects of it. And here, environmental surveillance, the modern methods using molecular biology methods is important because that gives you an idea about what is the health status of the living system in that particular region, right? So you can look at the bacteria, viruses. Depending on the load of bacteria, viruses, you can predict the health of the host. And you can also look at how host is responding to it using very different serological methods. If the titer is high or titer is low, and of course there are clinical data you can get, right? Which is difficult always to estimate. But whereas the simple molecular methods is easy to estimate quantitatively with some error bars, but that error bar will be very narrow because you are actually very precise when you look at if this is the titer of the IgGs, for example, against let's say FMDV. It's unlikely that that particular individual animal would have had severe disease because the immune response has been pretty good and so forth, right? And of course, IgG is just one example. It has to be a battery of tests. The second one is the population size, right? We can use variety of different, particularly for forest or for large animals. You can use variety of methods for doing population census of those animals. That also an indicative of the ecological situation. And using that, you can actually have what is called One Health metrics for each region. Recently, a friend of ours, a neighboring institution, Rakesh Mishra was earlier director of CCMB here. He is now the director of TICS, and they have developed what's called One Health metrics. So, for example, based on certain parameters, there are about certain parameters, you can say what is the One Health indicator for that particular region. And then you can actually, like human health, what's called human happiness index, human welfare index. There are so many methods people are using, the sociologists. Here also you can use using modern scientific methods, which basically gives you a true situation in terms of what is the One Health index for that particular region. And then you can work it out what actually you need to do. If it is, let's say you ideally should be 8. If you are 6, what you need to do to go to 8 and so forth. Now, coming to our initiative, it all started with the COVID. Many of us got into so-called public health, got into use of our expertise and our techniques, our tools in the lab for public good. Of course, whatever we do in science is always public good. No science is, whether we do basic science applied at translation, translation is closer to the public good, applied is nearer to the public good, basic science is slightly far, but everything is for public good. But immediately, what we are providing service to the society happened because of the COVID, right? I'm sure all of you know in March 2020, when the COVID hit, we had 2 testing centers for the whole country, one in Delhi, one in Pune, and National Institute of Virology, right? And within a year, we had 12,000 testing centers, and large number of them were run by academic people. In IISER Pune, we set up one. The head of the testing center was a plant biologist. because we as scientists, we know there is no difference between, an RNA or a DNA, what you do, PCR, RT-PCR, whether it is from bacteria or from plants or animals, right? So it's for us, it's an RT-PCR reaction, and we know how to reproducibly do a good RT-PCR reaction. And we RT-PCR machines are the same, whether used for diagnostic purposes or for research purposes. So hundreds of or thousands of RT-PCR machines across the country were repurposed for COVID diagnostics, right? Some of us who, some of the institutions who are not doing RT-PCR testing their own way, they donated their RT-PCR machines to a nearby diagnostic centers, right? That's how we served the society immediately at that time. Okay? Because otherwise testing and isolation was a problem. If you compare to the rest of the world, when we started testing, as I told you in May, June 2020, people had to wait for 2 weeks to get the testing results. A friend of mine, my classmate, in a small town in northern Karnataka, he had some kind of a symptom. Doctor said, get isolated and send your samples. Sample had to go to Bangalore and come back for testing. It took 2 weeks, and yet later it turned out he was negative. Unfortunately, unnecessarily he was in prison for 2 weeks, and and turned out to be negative. But later, within 6-7 months, our turnover was so good. India was the only country compared to US or even UK where you could give your samples in the morning. You don't have to stand in the long queue. You can get results by next day morning on your mobile phone. Everywhere else, they had to wait for 2-3 days to get an appointment to give the samples, and then another 2-3 days to get the results. The 6 days minimum they had to get isolated without knowing whether they're positive or negative. This happened because the community joined to respond to the COVID pandemic, and large number of them happened to be people like us, you and us, who knew what is RT-PCR and how easy one can do this. And what we need to do is to take care of it, because as a researcher, you can say if this PCR didn't work, the principal will say, go and change the buffer or do this, and you will get it. The second thing we did was, with this, I don't know if anybody is from Sapala or Howell, with Hyderabad-based companies are here, is developing kits. Once we had a lockdown, why I'm telling you is for environmental surveillance in One Health context is very, very important, is making these kits and making it affordable and accessible across the country. Otherwise, we cannot do any, environmental surveillance for any of the livestock diseases or poultry viruses. So what some of us got together, people like Satyajit Mayer, myself, Taslim, Rakesh Mishra again from CCMB, and some people from NCL and ISA Pune and so forth, is to put a consortium of academic institutions and say, we will give you free of cost testing, validation, or any other scale up help that you require to make very different components which go to a testing kit, RT-PCR testing kit. This is still working. This particular consortium is still working. There are 100 companies and large number of academic institutions, and they ones who look at DNTPs, amidites, if it is Tachman probe based RT-PCR, and also all kinds of other reagents, very different plasticware you need for developing the testing kits and so forth. And this consortium helped us to generate close to about 1 million kit per day during the Delta wave in 2021, right? And cost came down because, as you know, initially we used to pay 2,500 to 3,000 rupees to get one test done, but later it came down to about 500 rupees within 1 year. It's because this consortium made close to about 1 kit per billion per day, and increase the production and reduce the cost, right? So that basically made complete indigenization of RT-PCR testing kits. And this consortium is going to help us in developing very different environmental surveillance kits, whether it's for FMTV, LSDV, or for avian flu and so forth. And during that time, we also started, in addition to genome surveillance, we also started what's called, wastewater surveillance, right? And basically, the humans who are infected, whether they're symptomatic or non-symptomatic, they shed virus. If large number of them are asymptomatic, you wouldn't know whether the virus is spreading in that population or not. And so if you do wastewater, the domestic wastewater that comes, whether it's through STP or in general open drain, you can pick up that sample, you can test for the presence of the virus. Depending on the viral load, you can predict how many people in that particular region may be infected. Of course, you need certain mathematical modeling for this based on the clinical prevalence, some of the earlier data and other things we need to compare, right? So we started, and I can show you one example. Look at this, right? Now, in December 2021 onwards, December 2021 onwards, they're already post-vaccination. There was Omicron variant had come. Many people were already well resistant to this. Hardly any cases in the city. So that means there were no clinical tests were done much. There were no clinical reports, right? Yeah, the red line is for the clinical incidents. Look at this. This is the highest number of people who had infected with SARS-CoV-2 compared to any time during the COVID pandemic, but hardly any cases. Of course, this is the success of the virus. It tells you, success of the vaccines, right? Almost all the vaccine skeptics, just looking at this itself should feel that there are two things. One is environmental surveillance is the best way to understand the prevalence and try to take preventive measures. Second, if there are any people who think there are skeptics of vaccine, you should show, you should go to schools and colleges and tell them, show this for our general public. Look at this, the amount of virus that is present, hardly any incidents, simply because of the vaccine. Okay, I'll skip this. Now, what we're doing is COVID-19 beyond the second wave, and then we want to sort of get into other pathogens, viruses. We are doing now wastewater surveillance and the genome surveillance for very different types of influenza viruses, dengue, chikungunya, and also AMR, antimicrobial resistant bacteria and so forth. Basically, what we are doing is standardization of the protocol, work an entire workflow, and developing the kits along with our industry partners. Now, what we are now made a NCDC, National Center for Disease Control in India, which basically monitors very different protocols for this. NCDC now wants to send out a circular to all the public health organization in every state that they should take up environmental surveillance in major places. Of course, entire country is impossible. Some like railway station, bus stand, airports, nearby, hospitals where large number of people congregate. And that places, if you do environmental surveillance, and we have now set up a joint committee between NCDC and our consortium and developing all these protocols, which are like do it yourself kind of a protocols along with the kits, and we can provide this. Of course, kits will be directly supplied by the companies because they are the ones who are manufacturing and selling the kits. But we are working with those companies. Huel is in Hyderabad is one of them. And those kits will be affordable at low cost because their cost of R&D is also low because we have provided free of cost many of the services, R&D services. Sorry, I'm going a little bit faster in the interest of the time. Basically, I'm telling you SARS-CoV-2, we still continue in case some new variant comes, which is going to have some, let's say, vaccine escapers. So far, thankfully, we have not had a single vaccine escapers. Second is the surveillance for AMR. AMR is another major burden. AMR is also a problem for other animals, not just for humans. And of course, dengue is a major public health problem in this country. And also many of these surveillance also helps to deploy vaccines. In fact, I'm going to come to that in the context of, I think I'll skip all of these things. Okay, now we have just initiated a new project going beyond human pathogens to animal pathogens, particularly for foot and mouth disease virus and lumpy skin virus of the livestock, as well as the avian flu of the poultry, right? Workflow is more or less the same. You take very different types of environmental samples, which basically could be dung or could be slurry, could be water flowing out of the holding area, or it could be milk, or it could be even air. For example, avian flu, we have a successful in detecting the virus in the air from a nearby region. Or in case of One Health context, if many of the avian flu also sort of goes back and forth with domestic versus wildlife birds. You can also go to, these, lake beds and collect the samples. And it could be the bird drops, or it could be, air itself in that region, if the large number of birds are congregating. And that also we can be used for monitoring the presence of avian flu in the wild birds. And all of them are using, we look, collection of samples look, very odd, it's a simple, straightforward, but it's still deep science, where to collect the samples, how frequently you collect the samples, and how to correlate to the clinical prevalence. All of this requires good statistics and understanding of the, very different the correlation factors and so forth. Once it is done, and these samples would be, processed and standardized. So finally, for actual routine surveillance, with the help of ICR institutions, we may use, let's say, only air for one particular type of air, or only water, flowing water, or simply water trough. For example, many times when multiple animals drink from one trough water, there can be mixing of the saliva, and then virus may transmit between the animals. So that could be where one way is to just use the water trough for collecting the samples and do the surveillance. And for avian flu, of course, it should be a complete, because there is a lot of possibility of transmission to other wild birds, and particularly like waterfowl and so forth. And this interface we need to be looked into, and then do the surveillance in these kind of ones. And we have had some good, sorry, I don't know, some slide is missing here. Sorry, some slide is missing. Actually, we had some results of surveillance. I think I'll skip this. Just want to mention this a little bit. I'll tell you why. Okay. Anyway, so this is the kind of work we're doing. Some slides are missing. What we are doing now is to do the surveillance in certain specific areas where the low, medium, high clinical prevalence of FMDB, LSDB, and avian flu. And then we do environmental sampling and do this surveillance for about a year across all seasons. And during this time, we can actually, data that's collected can be used for mathematical modeling to develop an early warning system. A disease model is developed, ecological model of the disease, and that will become an early warning. Why it's called ecological model? Because you also can consider a variety of other factors. We don't only, for example, in human, many times a disease model, we only look at our immunity and physiology and the viral virulence, right? Whereas if ecological model, you have to look at temperature, humidity, what other factors are there, and mobility, and variety of different factors you have to look into it. And that will become an early warning system, which can be used by the animal husbandry programs, where they can actually, where vaccine should be deployed on priority, where animals should be isolated on priority, and so forth. That can be looked into. And a last thing is, just in one way, beyond health related to infectious diseases, other way is also that because of the climate change, we also need to, invest a lot of time in developing climate resilient methods for animal welfare, right? And otherwise, we do whatever we do, it's only look at from the human perspective, it may not be useful for other animals, particularly wildlife. And variety of different ways the habitat is getting degraded or defragmented, forests getting fragmented or deteriorated, and that will have an impact on the animals which are dependent on those forests. Forests can be two types: tree forest and also grassland, like savanna and very often in Indian, as per the law, and only recently they changed it. Earlier, during British time, a forest is considered where there are trees. The grassland is called wasteland or barren land. So only post independence around 1970s, Indian Forest New Act at that time, where the grassland was considered as a natural habitat for animals. So that also is a protected region, right? And if you look at what happened, I'll skip this one last slide. In Uganda recently, because of climate change, large part of the forest in which these mountain gorillas were living were getting degraded, and there was mountain gorillas became really endangered because of lack of availability of nutritious food for them. It turned out to be that one way to replace that kind of a habitat is bringing lots of bamboo, because also bamboo is also part of agroforestry. So it's a community forest were developed, and it turned out to be that the shoots of these bamboo turned out to be a good diet for gorillas. Now gorilla population is increasing, so in a way it's a win-win situation for Uganda. In fact, visited this few months ago to discuss some of these programs. In fact, in my first slide, I showed in Animal Day all the pictures of zebra, elephants, and lions. They're all from my visit to Uganda. Okay, so I'll leave this. Again, I have already spoken a lot about what we need to do at the public health perspective or One Health perspective, what an organization like ours can do it. We are all molecular biologists, we are lab biologists, we use our expertise, and all of these are important. Samples may come from an animal holding area here, or from a mouse lab in your animal house, or may come from some other roadside sample. Doesn't matter. Finally, your molecular biology is you isolate nucleic acid and look at what it is, right? So all of us can contribute to this kind of a program. I'll stop here. Thank you very much.
Thank you, sir. Thank you so much for your insightful talk, which underscored the importance of the critical role played by environmental surveillance in maintaining the ecological balance. And also cater the need of the earth. So, we will take some questions, please.
Yeah, that's
So this environmental surveillance, is that a project at NCBS or is it?
yeah. It's a LCBS is only an anchor. It's a network. Actually, that's what one slide is missing. There all the pictures are there. So GBRC, Ahmedabad, Gujarat Biotechnology Research Center, National Chemical Laboratory in Pune, Nivedi, Nishad, NFMDB in Bhubaneswar, and then we also have TICS, Tata Institute of Genetics and Society in Bangalore. So this is a consortium, multiple institutions, and working with a mission mode. So we have program managers who are domain experts, but they are the ones who keep managing the flow of information, data, money, partnership and everything.
This Tata Institute is also Rakesh Mishra.
Rakesh Mishra was also involved.
Yesterday he was
Yeah.
mentioning about it.
Yeah.
At GST.
Farah Itiak in his institute. She's a, wildlife disease expert.
Sir, I have a question. A wonderful talk and
Thank you.
a different perspective, the ecological aspect. So recently, NIV had a program called Milan, where we have multiple stakeholders meeting in a particular region. This was in VCRI Namakkal Veterinary College, where I happened to interact with one of the farmers who had a very unique question to me. So he just noticed that I'm a scientist, and then he approached and he said, I have a problem in the village where I stay. Previously, they were growing millets. And then now a lot of people have migrated to city. So people who are staying there started growing only trees. So within a span of 5 years, the entire village is covered with trees, and because of which it has attracted a lot of peacocks. So the peacock population has increased so much that it has become a menace right now. So clearly there was a disturbance in the ecology there, and he was asking whom to approach and what could be a possible solution. So I had no answer for him, but I said I will, go back and check.
Well, I mean, in many countries, what they do, even in India also, there is what's called culling, right? In fact, whenever the deer population increases so much, that will sort of imbalances the ecology so much that it has to be culled. It is allowed in the it's done in other countries too, right? So whenever some animal population increases because of some human activity imbalance, whatever it is, then the word is culling. Or sometimes they transport them to other places, so that of course you don't want to kill them, you want to sort of move them to another places. So there are variety of different methods are there. Wildlife management people, they have a proper procedure for these kind of things, legally validated procedures.
Also, I was interested to know if there are any ecology balance policies Like, I don't have to grow so many trees in it, because
Is
nowadays.
there, it is there, depending on the niche. For example, some time ago in Andhra, they were trying to convert all the grassland into either forest land or to convert into agriculture land. So many ecologists basically protested. They came out with a policy that it should not be done. It was about 10, 15 years ago. And finally, they agreed that some land should be retained as grassland. Of course, but because of developmental activity for other reasons, many times we convert, a natural land into something else, like human, some, either residential or manufacturing or development purposes or putting a road or something. So now they are considering all of these things. So they have an expert group of ecologists, because that's why, right, without environmental ministry's approval, they cannot take up any of these kind of developmental work. And with the ministry, what they do is they use some experts. Experts can be within the system, like Forest Research Institution or Wildlife Institute of India in Dehradun, or sometimes they call people from, our NCBS people like Umar Ramakrishnan keeps going for these kind of meetings, or Sukumar from Indian Institute of Science. They do advise government.
Second question is on the environmental testing that you do. Generally, from a clinical sample itself, when you do testing, sometimes the test doesn't work. So what is the process that goes behind to ensure that the environmental?
There are two reasons. The testing is depending on the sensitivity and specificity of the kit, right? So one of the things we have been able to do it for COVID and also for dengue, also, and for AMR and for environmental sample now, is also increasing the sensitivity of this one. Even if one or two viral particles are there, you should be able to detect it, right? And one of the things required for this testing kit to standardize such kind of sensitivity is it's a lot of R&D, which also uses some kind of standards. Very often use synthetic RNA or if it's RNA virus to test a kit under development. But what we have done is we have developed, almost not manufacturing, growing large number of cells and extracting viruses from those living cells. Those viruses we purify and get large amount of pure whole genome mRNA, which is used as standards. Increase the sensitivity of the kits. Of course, there are other problems. There are many times when you take a DNA RNA from environmental sample, typically also come with other contaminants which inhibit the enzyme activity. So purity of the nucleic acid is important. So you need to test very different methods of purification. Sometimes PEG method, sometimes other methods and all. Some of the methods are very expensive. We cannot afford for routine surveillance. So we're also trying new methods of nucleic acid purification.
Builds the protocol for even the sampling process.
Yeah.
What I'm seeing, of this One Health for the where how we are addressing this cultural and societal challenges. In this One Health project, one of the challenges is this also cultural and societal challenge. In like a country like India, where we are having many societies.
Yeah, it's maybe where sometimes it's a problem of social scientists will be able to better address rather than scientists. Typically, we are good in treating the symptoms rather than the cause of the symptoms, like all doctors do, right? In human doctors, animal doctors, everyone. The reason is it's easier to do that. Environmental surveillance at least helps us to tell where the root problem is, how to prevent this outbreak and everything. But as you said, a lot of it is also cultural, right? For example, during the large LSDB outbreak recently, is also contributed because of cattle on the street. There are so many of them on the street and unmanaged because they are left to the street. And that in the last few years, that also, add to the spread of the virus very quickly across the country. Now, this is a political decision, not a scientific decision you can deal with, right? You should not leave cattle on the street, right? Then how do you manage this, right? So these are the things which for a scientist very difficult to address.
As a
maybe a bit embarrassing, but no, you were just mentioning like, political decisions, right? As scientists, we need to, are we not ready to highlight this and then impress upon the politicians to take some decisions? Like, one thing is like, in the earlier part of your talk, you were mentioning about, say suppose in a forest ecosystem, like when the carnivores are going to be on the rise, like, it is going to kill and eat away the herbivores. And so we try to maintain ecological balance. I personally feel like, for the entire ecological or environmental disturbances, if not in other part of the globe, at least in India, is because of the explosion of exploding human population. We have been concentrating on that some 40, 45 years back, like you might be knowing, like we had lots of programs, like. But of late, we are keeping silence on that. And is it as an ecologist or as an environmentalist?
No. So, okay, I understood what you're saying. It's talking about the human population, right? So before this one, I know mentioned that I'm not, I mentioned this as a political decision is we are talking about this in all aspects. We generate data, we tell the people, we tell the government officials that, this data clearly tells you. For example, COVID-19, we all got together, look at all the data, try to see how the disease spreading in different parts of the country, tell the people that, government that you should do this. Sometimes they listen, sometimes they didn't listen. So people died. Of course, we lost, quite a bit, quite a number of people during the COVID, but somehow we managed because of variety of other measures that brought in both the government and by the general public. Although we are government employees, we are not mandated to do all of these things. We contributed as if we are knowledgeable citizens of the country. Coming to the population thing, but all the statistics says whatever is the growth has happened post green revolution because of availability of the food and nutrition, food security, and that's a lock phase we reached. Like any other country can go through this once in a while, because we started with huge famine, under malnutrition, and everything. Mortality, child mortality was very high. The woman mortality during pregnancy was very high. All that was stopped because of better healthcare and everything. And suddenly we went to this one. And 1970s, that's where we are taking the benefit of green revolution and public health, programs of the national level. 70s and 80s, there was a big push for family planning because people knew that is unbearable, unmanageable with increased population. Why that kind of a drive is not there now is mostly because those are the time when literacy was still low. 70s, 80s, if you look at the gross enrollment ratio was less than 10%. Now it is 25%, with 26 or 27%. We are not, but still a large number of people are well educated. But if you look at education is the one which is basically reducing the population size. So more emphasis on education is automatically reducing it rather than forcing them to, for the family planning. Yeah. So, in that way, if you look at what happened in Gujarat, I mentioned that one. There's also a political decision, which was highlighted by a scientist, that you should have allowed Gir forest lions to have a migration corridor with Madhya Pradesh forest. So the government said, it's the pride of Gujarat. We are not going to share the lions with Madhya Pradesh. But they lost about 200 of those lions because not allowing the migration corridor. But at least those would have survived. These are the kind of political decisions happening in the country, again, often and on in different time. Scientist's job is to highlight what happens if you don't take right decision. One can become an activist and do a fasting or something, but I don't know. Personally, I don't know. Sorry.
So yeah, we are in a serious thought to that for the future. Anyway, thank you, sir. It was a very interesting lecture.
Thank you.
No further questions. Thank you, sir. Thank you so much for your time. Thank you. I will request our director ma'am to kindly felicitate Professor Shashidhara sir with a memento and as a token of gratitude from us.
Thank you, ma'am. Thank you, ma'am. As we conclude this event, celebrating World Animal Day, it is my privilege to extend our heartfelt gratitude to all those who have contributed to the success of today's event. First and foremost, we sincerely thank our director, ma'am, for her unwavering encouragement and support, which continue to inspire us to strive for excellence in all of our endeavor. Thank you, ma'am. A special word of gratitude to our chief guest and speaker for today's event, Professor L.S. Shashidhara, for graciously sharing your valuable insights and for dedicating your time to enlighten us today. Thank you, sir. We're deeply honored to have you here. I also take this moment to thank Professor Redenna for joining us today, making this event even more memorable. I would like to extend our gratitude to all the distinguished speakers and esteemed guests in the audience and graciously joining us online, whose presence has added great value to this occasion. We are deeply grateful to the heads of various sections of our institute: administration, finance, stores and purchase, engineering, ICT, security and housekeeping, whose efforts ensured that this program was executed flawlessly. A special appreciation goes to our colleagues and students of NIAB whose enthusiasm and presence made this event even more successful. Last but not the least, I especially thank the members of Scientific Event Management Committee for their efforts in coordinating and organizing this event, leading to its seamless execution. Thank you all once again and for your participation and support. Thank you. Now I request everyone to please gather at the podium for a group photo. Please gather at the podium for a group photo. Thank you. Thank you.